they would have the similar questions. Okay, so I've got the local recording going on, and I guess I should try and do the Zoom recording at the same time. All right, so usually when we do regular old lab, I like to always before we dive in, I like to go over the pre-lab with everybody because you know, I don't expect you to get it 100% right, but I just like to see how much effort you put into it. But having said that, of course, we need to make sure that we know what the right answers are, so that we don't go away from the class with confusion or wrong ideas. So I think that I gave you this introduction, I gave you an objective, and the bottom line objective of this class is this lab is to understand how statistical methods can be used to analyze data. It's really in its most simplest form. It's kind of weird because they go through this whole uh, talk about this one-celled organism and all this neat stuff about circadian rhythm, but you actually don't get to do any of that. um, it's in 1200 lab, especially if you take it with me. There's a more interesting one where we actually do take data of people's blood pressure and an EKG of their heart. So we take our own data, but then we analyze it in a different way. It's data that's different than what you might be used to if you work in a hospital and you've simply taken blood pressure or done an EKG, but it's a way because it's called physics of the cardiovascular system. And bottom line is, after doing the lab several times, I finally figured out that the physics part is the data analysis. And whoever wrote the lab, well, it's old school. Back in the day when I took classes like this, the philosophy is that they just taught you physics the way physicists know about it, and it was up to the students to try to figure out a lot of the gobbledygook. And I don't believe in that. I believe in breaking things down into small ideas for you guys and then trying to give you context as well, which whenever I used to ask one of my professors, how does this piece of information fit in with everything else, they usually, worst case, got mad at me. Second case is they just didn't answer. And I realized over time it's because they didn't know. And maybe it's just because they didn't spend any time thinking about it. But I don't know. I'm the kind of person I think about everything. It kind of annoys my wife sometimes. So, anyways, let's run through the pre-lab first. So, the first question is kind of open-ended. I went online and got some better pictures than they had, so it's a little bit easier to see. Um, I didn't notice. I think that these are neat. People use them probably because they're large, but they're still single cells, relatively large. Yeah, 30 to 40 millimeters long. That's pretty big for a cell. So you can study things about single cells quite easily, and it is kind of cool looking, I suppose. I mean, and there's no. It's pretty open ended. So circadian rhythms. This is a word I will never forget because I took the biology AP exam in high school, but I just took it on my own. I just registered for the exam and I didn't take a class. I just studied on my own. And the very first question was what time periods do the circadian rhythm approximate? And I never even heard of the word. And I said, oh crap, maybe I made a big mistake not taking in a biology class. But I ended up doing all right on the exam. But it was pretty funny. And I was just saying, I think you screwed up here, Rabbit. Okay, so circadian rhythms are biological time periods. So a period is like a sine wave, like one wavelength. So anything that repeats itself has a thing called a period. So, for example, your art beats so many times a second. So you can invert that to tell you how many seconds per heartbeat. So, and then let's see, circadian rhythms are basically, I believe, only for biological things. And I also noticed that it's usually only 24. It's usually 24 hours, and usually associated with light and dark cycles as well. So I think I'm not happy about this. I was just looked it up in Wikipedia because, in fact, the experiment they were looking at, they didn't turn the light off ever. So of course one could test that, right? So it's basically about rhythms of life, if you will. So many people may know about the 28 days of the moon and 28 days of another rhythm that maybe I won't get into too deeply because I don't want anybody to be offended or embarrassed. But I'm sure somebody out there knows something about it. Okay, so how, and I forgot to say why, was the data taken? So what was done is, and I have a picture on the next page, that the transmission of light through the cell at one end was measured, and you could track chlorophyll, well, chloroplasts, the things where the chlorophyll is in, and you can imagine if there's more chloroplasts in the way, that some of the light's going to be blocked. And there's certainly refinements to this. You could use different color of light, but as far as I can tell, it's white light, and it's using the I put experimental form, so it's using the adult form, but before it starts to try to reproduce, which I don't know why, but that's what they picked. And so the idea here was to see, because if the circadian rhythm was dependent on the cell having a nucleus, for example. Because if you kind of think about the nucleus as it's not exactly the brain of a cell, and there may be people out there that could do a better job than me, but the way I think of the nucleus, it stores RNA, right? And so when the cell needs to create new proteins, it uses the RNA in the nucleus. Then there's a transcription RNA that makes the mirror image, and then the transcription RNA goes out to the cell and assembles the amino acids, and you get the molecule you want. And so the nucleus is sort of like the hard drive. It stores all the blueprints for everything that has to go on in the cell. So one could imagine that maybe if you remove the nucleus, which is the rhizoid end, that it might affect the circadian rhythm. Might even stop it. Who knows? So you get your cell, and you have the detector just here in one place, and you have a light. So apparently somebody had noticed that the chloroplasts would go back and forth over 24 hours. So you imagine that when they're up here at this end, this is when you'd see brightness, and then when they're down over here at this end, the let's call this place 1 and place 2. So these tops are 1s, and the bottoms are 2s when the chloroplasts are all down there. And as I said earlier, the light source is always on. So I guess somehow there's been some kind of control done for whether or not you need, because before, like, the simple definition of circadian rhythm was that you're supposed to have night and day cycles being involved in it. And this experiment certainly didn't have those things. So anyways, you see here. Oh, sure, sure. Yeah, huh? That's yeah. Yeah, right, right. So that's a good question, right? Because I know, as far as I understand it, human beings, if they're kept in a room where they don't they leave the light on all the time, or not turn, or leave it off, that drives humans crazy. But it becomes difficult to understand what time of day it is and how many days you've been there. Oh, I agree with that point. I'm not an expert. Um, but apparently, and it might be just because we're a higher form of life, right? Studying biology and being quantitative about biology is tough, and it's with the advent of high-powered computers that it gets more and more quantitative. But in this case, you study a simple organism and you hope to learn something that you might be able to apply to higher order organisms. That's the best I can do, and I can't really answer it completely correctly. Yeah, the lab manual needs to be rewritten in that sense to say, look, this is an approximate definition, and because this lab is very open ended, right? We could spend years on every part of it, so which I'm trying to focus it down so you guys can take something away from it. But we still have to have a certain amount of background to understand what we're doing. So, anyways, you can notice that this is taken over many hours, and if you were to sort of look at the places where the waveform repeats, you would probably notice that it's about 24 hours. But this isn't really good enough because it's difficult to assign an error bar to that. And that's one of just as a side note, that um, um, hard to define, uncertainty is meaning, like the plus minus on a measurement is the reason for this kind of analysis. And we'll get back to that. This is sort of like an advanced organizer. The reason for stat analysis. So, how do we get? So the next question is, how do we get from that to the data that we're about to work on? So they say in the manual that it's a pretty tough thing. Oh, my numbers aren't staying with me here. Let's see if I can make this a single. So cut, paste. Now they'll all move together. Okay. So how do we extract the data out of it? And it's a thing called a Fourier transform, and it's heavy math, and it's way beyond really any freshman class, and especially a physics 1100 class. But what you notice is that, first of all, you kind of see this sine shape, right? This gross sine shape. But you can also see wiggles in different places, huh? And these wiggles have different time periods. In fact, if you look, there's also another one here, like that. So there's some other really long time period. So what a Fourier transform does is it takes the intensity as a function of time. Why aren't you working? And it changes it to intensity. Well, basically, it changes it to be very fair. I put time period here, but when you do a, I don't know what the how you do Fourier symbol, but if I Fourier transform from time, what I do is I Fourier transform into frequency. It turns out, but frequency. Remember, I said like your heartbeat is a frequency, so number of beats per second. So the period is just the number of seconds per beat. How long does it take to repeat? So I take this time data and I transform it into period data. So these would be hours. So you can see there's little wiggles. So you would see something like this. And then we notice this long period. So there might be something, of course, way out here, because that looks like hundreds of hours. So we're at 25, 30, 35. So way out here, you might see another little peak, because there aren't too many of them, right? But what you would see is that there would be one, say 24 is right about here, and we'll just assume that the dominant frequency or period is 24 hours, but you would see a peak like this. And so I did a lot of studying to try to figure out, searching to figure out how this is actually done, because they give us a bunch of numbers later on, and what I gather is that they break up data like this over hundreds of hours into, well, if you broke it up into 24 hours, that wouldn't be good. So I guess that at the end of the day, to get the table of numbers we're using, where are they? Here it is. That this is like several of these like 300-hour periods, many of these 300-hour periods. As I don't really know. But what happens is then, and this is also statistics, it's pretty funny. Notice that this P here is centered on some value, say 24.8, and then this has a certain width. So if any of you guys may know about what's called a normal curve or a bell curve, and you see that in students' grades, grades and IQ plots. What about heights? People's heights, or the heights of average height of a man in the United States. So this is called the mean, the average, and this is called the total standard deviation. And so what happens is the Fourier transform program, it sees, so over here is like 24 maybe, and up here is 25. So it plots all these points, and then it says, okay, how I want to put a line on this, a curve, and I say, I need a standard deviation, I need a bell curve, and it adjusts the mean and the total standard deviation so that the line fits on the data points. And again, I'm trying to give you enough information so you understand it, but not too much that you don't lose your mind. So what it seems like is that the table we have are this Fourier transform telling us what is the mean. So let's just say that we take, I don't know how many numbers we have. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15. So you take 15 300-hour plots. And for each one, and time plots, and for each one, you do a Fourier transform, it's called, and you find out what's the dominant frequency, period, what's the dominant period. This is not super important to write up in your lab manual. It's basically just these things I typed in here. I'm just giving you a feel for the background so that you kind of understand what's going on. Well, I'm trying to put it into context is what I'm doing. But I don't expect you to be able to quote me and stand in front of the class like and say what this is, all the stuff I wrote down and read. So the bottom line is that we take a time plot, we transform it into an intensity by time, we transform it to an intensity by period, and then we look to see what's the dominant frequency period, and then we just say what's the this highest point. So we find the highest point, and we say what is this x value from the highest point. And we did that. In this case, we did it 15 times. And it was done 15 times for each of the two populations. Remember, one population is with rhizoid or nucleus, and one population without. Vegas, huh? Where did that come from? Okay, so then it asked you to define some terms. So I had provided you with, let's see, well, I thought I downloaded it. Where did it go? Ah, right. So I provided these slides, and I provided you a recording I made last year. It was physics 2100, I guess, but this part is pretty much applicable. So yeah, so this was like there's there's a famous guy, Mark Twain, said that there's lies and there's damned lies, and then there's statistics. So part of what you're learning here is how to determine whether somebody's bullshitting you or not. So we say that, well, Obama surges past McCain because it went from a 2-point lead for McCain to a 2-point lead for Obama. But then you say, oh, well, it's plus or minus 4%. So that just, and this was a change of 4%. So that's called a statistical fluctuation. So that's basically what a statistical error is. Whenever you do, when your data is a count, then it's a little more than that, but for the time being, when your data is a count, you were talking about how people are in. Oh, yeah, right. That's exactly. Oh, yeah, tests, right? I was just reading about, I remember when I was a bartender, you had to empty ashtrays right away so people couldn't see that. It showed you my age when I was young and people could smoke in a bar. So we had to dump because, someone could see how long they'd been there by how many cigarettes were in the ashtray, right? And then the clock was always put so it's hard to see. Yeah, no clocks, and it's all right. Exactly, right? Very good. Good for you. Yeah, it's a kind of social engineering. Memes nowadays. Memes are like pictures people put on Facebook, but a meme is, the basic definition, is sort of a unit of an idea, and it's used in social engineering. Anyways, let's see. So anytime your data is counting things, or in what we're going to see today is combining numbers. And so that's counting things in a way. Counting, oh yeah, counting or averaging, say. To get a plus minus, we need the D plus minus is defined as a statistical error. Statistical error. Sometimes you don't have these things, but sometimes you do. Now, a systematic error is, this one's fairly easy to understand. It means that there's something wrong with your apparatus, or with the way with you. I'm just keeping this short, meaning somehow you're not reading it correctly. And a simple idea would be many rulers, right? Because they're trying to make them last longer, they put the 0 here. And so if you forget and you use the end of the ruler instead of the 0 to take a measurement, that would be considered a systematic error. A very famous systematic error happened, well, it must be close to 10 years ago now, where this neutrino experiment in Europe was measuring the speed of light. They were seeing a violation of causality. So a neutrino was being emitted from, from CERN, and it was going to Gran Sasso in Italy, many kilometers away, and they were seeing the time of the event here. T here was before, so T at Gran Sasso was before T at CERN, and people are going, "Holy shit, we violated causality. Something traveled faster than the speed of light." And in fact, they published this, but they said, "Look, guys, we don't believe it either." But they went over this data for 2 freaking years, and they published the data because they wanted other people to help them. And it turned out that somebody went into a cable room, and they went up to some box, some measuring, some device, and it had a bunch of cables, and these were actually light fiber optics. And whenever you went into this room, you were supposed to make a log entry about when you went in and when you came out, and you were supposed to put in also what cables you touched. So somebody went in there, pulled a cable off, put it back in, but they didn't push it in all the way. And so because it's a fiber optic cable, it still would work. So now you have your fiber optic cable and you have your device, but now there's this space in here, and the light can still travel, so the device still gets the data, but there's a time lag put into it. And when you're talking about speed of light and you're measuring these neutrinos are traveling at the speed of light, that little tiny bit threw off everything. And that's in fact the reason why they had the logbook on the cable room to begin with. But that's a very extreme example and famous example of a systematic error. So accuracy is basically how close to the truth or the real answer, which is actually not rarely known, the right answer. That's the whole reason we go through all this, so that the whole reason we do all this analysis is so that we can communicate our results to other scientists, and that they will believe what we say, and they understand our possible errors. And usually when somebody publishes a paper and somebody doesn't agree with them, they'll say, well, what did you do about your systematic errors? Because the systematic errors will throw off your accuracy. And then precision is basically the number of sig figs. And this is can depend on if you have statistical errors. It depends on your technique, and it depends on your instruments, and it depends on design as well. So all these go in, and they kind of overlap each other as well.